طيب, I have to present the basic concepts as follow-up for the groups that present the same concept, basic concepts. They present basic concepts. We are make follow-up for what they present. So we have to know the forward and reverse genetic if we want to apply genetic in the laboratories. We have to know two concepts, the forward and reverse genetics. So what is the forward genetic? The forward genetic is concerned with the observable trait, or it concerned the forward genetic concerned with phenotype. That means it begins with observed phenotype and works backward to identify the genetics mutation responsible for that phenotype. The process of forward genetic is sequentially divided to sequence here, phenotype to gene, and then identify the or make mutagenesis, make mutation to the the organism, then screening for the mutation, then make gene identification. Example of forward genetics, classic example is the study of fruit flies, Drosophila melanogaster. They make a mutation to this organism by chemicals. Then they make screening for those with visible changes like eye color and etc. After identifying such mutation, they map the genes responsible for the mutation. Advantage of forward genetics useful for discovering new genes and biological pathways. Can identify gene with previously known function. Disadvantage of forward genetic can be time consuming and labor intensive. The phenotype may be the result of complex interaction between multiple genes, which can complicate identification. That means if the phenotype contain a lot of genes that influence this phenotype, these make this phenomena of multigenomics or complex interaction between multi-genes make this test forward genetic of this phenotype is very very difficult. Then the other concept is reverse genetics, vice versa of the forward genetic. It begins with opposite direction. It begins with genes. It begins with genes. The process of reverse genetic is gene phenotype. Researchers start with specific gene, either one they suspect is involved in a particular process, or one they wish to investigate. The process can be applied by knockout of the gene. The knockout means we have to make the gene unfunctional. Gene knockout or knockdown. In reverse genetic, scientists often use technique like gene knockout, which is mean removing a gene, or gene knockdown, reducing the expression of a gene to study the resulting phenotype. This can be done using technologies like CRISPR-Cas9 or RNA interference. They study the phenomena after knocking out or modifying the gene. Researchers observe how the organism's phenotype changes, helping them infer the gene's function. They can infer the gene function by knocking out the gene or knock down the gene. Example of reverse genetics, CRISPR to knock out a gene is in mice. to study the effects of genes on disease development or behavior. Another example is if knocking out a gene causing a mouse to develop a disease or lose a certain ability, it provides insight into the gene's role. The advantage of reverse genetic is allows for a more targeted investigation of gene function, and this is useful if if the gene have is entering in the multi-complex gene, as we mentioned in the reverse in the forward genetic. This is the disadvantage of forward genetic. It had been solved by studying the genes one by one. This is the advantage of reverse genetic. It allows for a more targeted investigation of gene function. That means we can investigate if the gene enter in multi-function. It can make effect in multi-function in the organism. Vice versa of the forward genetic. Technique like CRISPR make it easier and faster to study gene function in various organisms. The disadvantage of reverse genetics may not reveal all the rules of that means if the gene have a multiple gene components for its loose, that means if a gene have multi RNA, sometimes the gene have multi product of RNA depending on the transcription factor that have rule in this gene. We cannot reveal all the rules of gene. We can miss. We will miss the rule of gene. The gene have multi-function. We will not interpret the result of the consequence of knocking out the gene. If, as we mentioned, especially when the gene has multiple functions. What is the difference between forward and reverse genetic? The forward genetic starts with the phenotype and seeking for the genes responsible for this phenotype. Vice versa, reverse genetic starts with the known gene and determines the phenotype produced by manipulating that genes. That means if we want to know the function of the gene, we have to apply reverse genetic. But as we mentioned, the disadvantage of this method is we want to know previously the genes. But if we don't know the genes, we have to begin with forward genetics and applying mapping for this phenotype that we have observed in this organism. That means the two methods is essential in the lab. So we have to know some definitions. So we have the multiple definition of gene according to its relationship with DNA, as we know. We can define the genes as sequence of DNA or functional unit of DNA or independent segregating unit or location in the genome. All of these definitions are correct, but I want to know what I want to tell me, you, my colleague here, who hearing me right now, what's the best definition we can apply for genes. If somebody want to participate right now, please. What's the best definition of genes here? Is it number 1 or 2 or 3 or 4? Sequence of DNA or functional unit?
The first one, sequence.
sequence of DNA is right, but what's the best definition? All of these definitions are right. The independent segregating unit is definition according to Mendelian inheritance, as I know. Location in the genome is also good definition. Okay, let's begin. Let's move on. The key definition. This is one of the key definition. Allele, version of the gene. The allele is the version of the gene. The mutants, the altered version of the gene. The genotype, all the allele of an individual. The wild type, a defined standard of gene. You have to know another concept in genetics in the lab. Yeast in the lab. This is the topic here, but the concept is called replica plate test. An example of this test is conducting experiment to investigate the genes necessary to the arginine synthesis in yeast. The test is the steps for your experiment from first to last. I assume you start with a plate of wild type yeast cells. We begin with a wild type yeast. That means it can synthesize arginine. Then we have to begin with wild type yeast cells. Then we have to mutagenize these cells randomly. The step number 2, incubate all the cells in rich medium. Then we have to make replica plate. The replica plate is a type of technique that you have to copy and paste the colony of the plate. Then number 4, incubate one plate of cells in rich medium and one plate in minimal medium. We have to apply copy and paste of the plate in two medium or two plate. One of the plate is rich and one plate in minimal medium. The minimal medium contain only the minerals and less carbohydrate and amino acid. Vice versa of the rich medium. Then number 5, sequence the colonies that fail to grow in minimal medium. As we mentioned, if we make copy and paste of the colonies, we will see in the plates that some colonies appear in the rich medium and not appeared in the minimal medium. Appear in the rich medium and not in the minimal medium. These colonies that not appear in the minimal medium is the target for us in the step number 5. We have to sequence the colonies that fail to grow in the minimal medium. So if we sequence the colonies that fail to grow in the minimal medium, then we will know the changes that or the mutation or the loci of the mutation that appear in the new mutant yeast cell. This is further description of the steps that we apply in the test. Then we have to know some concepts in the lab regarding the recessive and dominant, or we can concisely say something about recessive and dominant. First, the recessive and dominant result of an experiment. Recessive means loss of gene function. Dominance means gain in function or new function. The inference or inferred relative to wild type. This is the concept of recessive and dominance is inferred relative to wild type. Then we have to know the wild type in order to know if it is recessive or dominant mutant mutant organism, dependent on a specific phenotype. So the recessive and dominant is the description related to phenotype, not to genes. We cannot describe genes or mutation in the gene as recessive or dominant. Just we have to make this description to phenotype. This is one example of recessive, and can mention it as example of recessive is hemoglobin. If we study the sickle cell allele, and if we describe it as HPS, the organism that have HPS and HPS for two alleles, we describe it as sickle cell anemia phenomena. It will appear this sickness in the organisms that have two alleles of sickle cell anemia. Also, if the organism have sickle cell anemia and another allele is not affected by the mutation, also have sickle cell anemia. This is an example of recessive recessive trait. Also dominant, malaria resistant, sensitive to malaria if the alleles did not contain sickle cell anemia mutation, it will be sensitive to malaria. And also, if we have organism have only one allele for sickle cell anemia, it will be resistant to malaria. Only one allele sufficient to be resistant to malaria. This is example of dominance. البوينت بتاعت partial dominance دي. اوكي. طيب ده اللي هو الابدومولوجي. This is the epidemiology of sickle cell allele frequency and endemic malaria, comparing to endemic malaria region. This from this two map here, we can't know the endemic the relationship between the endemic malaria and sickle cell allele frequency because as we can see here, the sickle cell anemia area is less than 20%. It will not apply. It will not give you the relationship between the sickle cell anemia and endemic malaria because the sickle cell allele frequency is more or less than is already very few trade in the African people. As as I understand. Then the last concept I have to present here is the complementation test. This is the example of one of the complementation tests we can apply in the laboratory. Progeny of cross between two mutants is wild type. This is the rule of complementation test. That means if we make cross between two mutant organism and we have result of wild type, that mean we have complementation test. So we can see here in example histidine histidine synthesis function is losing here in this organism. X mean this histidine function or functionally depending on multi-genes. So if we apply a number for this X, that means the gene number 3 is knocked out or knocked down in this organism. This is the concept here. We make the haploid genotype histidine X. If we say histidine X, that means we don't know the genes that knocked out in this organism. And if we make crossing between histidine X and histidine 3, losing histidine number 3, gene number 3 lose the organism, and the phenotype is histidine losing function, that means there is no complementation test. That means the histidine X is equal to histidine 3. Okay, if there is no equality between histidine 3 and histidine X, like here, histidine X and histidine 3 losing function, we will resulting in wild type organism that will result in that complementation test is the complementation between the genes to result in the mutation, the wild type is successful in this test. Technically, we can apply complementation test in histidine complementation groups. We call it histidine complementation groups. We make crossing between the groups, as this table says, haploid strain and haploid strain. Haploid strain, that means this organism from 1 to 7 groups have only one allele in its genome. The yeast can depend on haploid strain or diploid strain, as we know, the yeast can be haploid or diploid. Then we have to begin with haploid strain. We divide it to groups and we make crossing out between these groups. Firstly, we have to know if the groups are histidine minus, histidine X minus, and applying crossing out, if if we apply crossing out for the same group, it will not give us the wild type organism as we see here. But if we crossing out between two different groups, it will it will result in a wild type result. That means the complementation test is successfully complement between two different allele, or complementation between two different mutations in different loci or in different genes. That means complementation test is successfully appearing here, like this example, 2 and 1. So depending on this test, we have to divide these organisms 1, 2, 3, 4, 5, 7 to groups. So let's focus in this example. If we want to divide this yeast colonies, colony number 1, 2, 3, 4, 5, 6 to 7 to groups, then we will say that 1, 4, 5 is the group number 1. Group number 1 contains organism or colony number 1, 4, 5, and group number 2 contains 2 and 7, and group number 3 contains 3 and 6. So we can divide the colonies from 1 to 7 to 3 groups, and we call it complementation groups, depending on if they give wild type or not. So if there is no complementation between 1 and 4 and 5, that means this 1, 4, 5 are in one group. They contain the same mutation in same gene. That means there is no successful complementation between these columns 1, 4, 5, and they put this phenomena puts them in one group. The group contain 1, 4, 5, and the other group contain 2 and 7, and the other group contain 3, 6. This is the complementation group. So in this example, we have 3 groups. Group number 1 contain 1, 4, 5. Group number 2 contain 2, 7. Group number 3 contain 3, 6. So here is an example of question to understand the complementation group. If your lab mate isolates another haploid yeast, then 8 with phenotype histidine minus, she crosses strain 8 with strain 1 from your experiment and finds that the haploid daughter cell also have phenotype histidine minus. Which of your lab haploid strain are in the same complementation group as strain 8? Select all the apply. Then please, if somebody want to answer this question, please, you can participate. Which haploid
الريومال سايد الباترن الفيل جروبس
according to this example here, if we cross out strain number 8 with the strain number 1, strain number 1 with strain number 8, and we found that it gives diploid strain with histidine minus. So which group, or if we make a list of complementation group, what's the complementation group contained in this example? Anybody? Okay, if there is nobody, I will repeat the ask, or I can solve this problem in order to finish this presentation. If we apply crossing between 8 haploid yeast strain number 8 with strain number 1, and we found the diploid daughter cell also have phenotype histidine minus, that means the group, the same complementation group as strain 8 is 1, 1, 4, 5. That means the new complementation group contain 1, 4, 5, and 8.